In today's lecture, we're going to talk about the basic unit of all living organisms, the cell. Before the lecture begins, make sure that you've read the assigned chapter and taken notes on that, as well as looked ahead in the PDF of the slides for this lecture and made yourself an outline so that it will be easier to take notes during the lecture. During the lecture, minimize distractions by turning off any background noise, music, movies, TV. Listen in a quiet space and turn off all devices. Take plenty of notes, making sure to put the information in your own words as a way to maximize your learning. So today we're going to look at life's properties that emerge because of the collaboration of the internal structure of a cell. We're going to look at the cell and its internal structures, and we're going to cover two types of cells, prokaryotic and eukaryotic. But before we start with that, we need to consider how to study a cell. In order to study cells, we use microscopes that magnify the structures of the cell. Cells were first seen by Robert Hooke in 1165. He used a microscope to magnify and examine the cells from the bark of an oak tree. And a few years later, Leeuwenhoek visualized living cells under the magnification of a microscope. By 1830, compound microscopes were widely used. These microscopes had two lenses. Both magnification and resolution were improved. Structures only 1 micrometer in size could be clearly seen. The light microscope was the earliest tool of cytologists, biologists who study cells. It allowed the identification of nuclei, mitochondria, chloroplasts, and other subcellular structures within cells. Light microscopy is also called bright field microscopy because white light is passed directly through a specimen. Phase contrast and differential interference contrast microscopy make it possible to see living cells clearly. The phase of transmitted light changes as it passes through a structure with a different density from the surrounding media. Fluorescent microscopy allows detection of proteins, DNA sequences, or molecules that have been made fluorescent by binding to antibodies. Confocal microscopy uses a laser beam to illuminate a single plane of a fluorescently labeled specimen. Digital video microscopy uses video cameras to collect digital images. In transmission electron microscopy, or TEM, electrons are transmitted through the specimen. In scanning electron microscopy, SEM, the surface of a specimen is scanned by detecting electrons deflected from other outer surfaces. All these specialized approaches allow for the visualization of specimens. The type of microscopy that's required to study the cell can be determined by the part of the cell being studied. These figures on the slide give relative sizes of different molecules. All right, now let's start by looking at cells. The cell is the basic unit of all living things. Cells are constantly changing. There are unicellular organisms that consist of a single cell, and multicellular organisms that have specialized cells for different functions. Two factors restricted progress in early cell biology. Microscopes had limited resolution and the descriptive nature of cell biology. The focus was on observation, with little emphasis on explanation, but now cell biology is a huge field and is often combined with biochemistry, genetics, and evolution. In the early 1800s, scientists thought that tissue was the smallest unit of living things. In 1939, Schwann postulated the cell theory, that all organisms consist of one or more cells and that the cell is the basic unit of life. In 1855, Virchow added that all cells arise only from pre-existing cells. There are 4 properties of all cells. They contain genetic material, DNA. They contain ribosomes. These first 2 characteristics or properties are directly correlated to the central dogma of biology, That is, that DNA is transcribed into RNA within the nucleus of a cell, and that RNA is translated into protein in the ribosomes of the cell. Ribosomes and the other organelles that function to help the cell survive are suspended in a fluid called cytosol. And to keep all these organelles together, there is a cell membrane, known as the plasma membrane or a phospholipid bilayer. Now let's talk about prokaryotic cells. Prokaryotes are single-celled organisms. Two examples of prokaryotes are bacteria and archaea. Prokaryotes have a unique structure that aid them in their unique functions. So again, referencing the structure-function relationship. Some of these structures are flagella, which provide movement, thick cell walls made of peptidoglycan and glycocalyx, and fimbriae, which allow the prokaryote to attach to the surface of something or to another cell. Prokaryotes' genetic information is in one chromosome within a nuclei region, but not a true nucleus. All right, so we just talked about the prokaryotes. Now let's look at the eukaryotes. As eukaryotes evolved, they developed compartments that allowed the cell to grow larger and more complex. These compartments are called organelles, and they are bound by semi-permeable membranes that work with other organelles to carry out cellular function. For eukaryotes, there is not a nucleoid region like in the prokaryotes. There is one or more chromosomes, and they are contained within a double membrane organelle called the nucleus. Examples of eukaryotes are animal cells, shown in the figure on the top right, and plant cells, shown in the figure on the bottom right. The cell membrane is composed of a phospholipid in a bilayer. Remember from our lecture on biological molecules, there are phospholipids that have a hydrophilic head and a hydrophobic tail. These tails align in the middle, and the hydrophilic head are in contact with the extracellular and the intracellular or cytoplasmic side of the cell. This bilayer allows for proteins to be inserted for communication and for anchoring. It also provides a barrier to keep out unwanted molecules that could damage the cell. The membrane-bound organelles are one of the key differences between eukaryotes and prokaryotes. So what are these organelles? The nucleus, which we just mentioned, contains the DNA in linear chromosomes. In the nucleus, DNA is transcribed into mRNA, which exits the nucleus and heads to the endomembrane system. The endomembrane system, which is responsible for moving proteins throughout the cell, includes the ribosomes, where translation takes place to generate proteins from the mRNA, the endoplasmic reticulum, which is known as the ER, and the Golgi complex. The ER and the Golgi help process and modify as well as ship proteins. Then there are the lysosomes, which act as a trash can, and vacuoles, which act as storage, and these are also part of the endomembrane system. There are mitochondria, which generate ATP for energy use in cells during cellular respiration, chloroplasts, which generate energy through photosynthesis in plant cells, and peroxisomes, which digest proteins and can detoxify the cell. Finally, there's the cytoskeleton that gives the cell shape and structure, like the bones in the human body. Together, these complex organelles work together to carry out the functions of a eukaryotic cell. So finally, you can compare and contrast prokaryotic and eukaryotic cells using various characteristics. You can look at the size of the cell, the presence of a nucleus, the presence of organelles, the material in a cell wall, the presence of flagella, cytoplasm, ribosomes, DNA structure, and the process that they use for cell division. So as you look at this figure on the slide ahead of you, this will be a great way to quiz yourself on how well you know the prokaryote and eukaryote cells as you prepare for the quiz and test. So next, let's get into more detail on the internal structures of the eukaryotic cells that we just talked about. We're going to start with the nucleus. The nucleus contains the genomic material. There is some genomic material found in mitochondria and chloroplasts, but the majority of the genomic material is found in the nucleus, where it is arranged in chromosomes. When the DNA is not dividing or being transcribed, it is wrapped tightly around the histone proteins, which give the chromosome a more compact packing within the nucleus. The nuclear envelope is a double membrane, a lipid bilayer, that separates the nucleus from the cytoplasm. Because DNA is transcribed into mRNA in the nucleus, and that mRNA has to head to a ribosome in order to make a protein, there are pores that regulate the mRNA leaving the nucleus. The nuclear pores are shown in the inset image on the bottom. The top image shows an animal cell with a nucleus, which is made of the nuclear envelope, a lipid bilayer, the nucleolus, which contains the DNA. These structures, as well, are shown in the image on the bottom right. You can appreciate in both images the ER with the ribosomes attached, which is known as the rough ER, is located right next to the nucleus. This makes sense as we think back to the central dogma. The mRNA has to leave the nucleus and traffic to a ribosome to be translated into proteins. The close proximity of the ribosomes, the rough ER, allows this process to be very efficient. All right, now we've considered the nucleus and nuclear transport. Let's look at the endomembrane system. The endomembrane system is responsible for the synthesis of protein in the ribosomes, protein transport, metabolism, and detoxification. The members of the system are the ribosomes, ER, Golgi, lysosomes, and vacuoles. Ribosomes are the site of translation. They are not membrane bound, so they are not considered an organelle. Ribosomes are shown in the top image on the right in the rough ER that is adjacent to the nucleus. In the image below, there is a transmission electron micrograph, which shows small beads or black dots along what appears to be tubes. These tubes are the ER, so the EM shows the rough ER. There are also free ribosomes in the cytoplasm not attached to the ER. Ribosomes are made up of two subunits, the large subunit and the small subunit. Ribosome subunits differ in bacteria and eukaryotes. Often, antibacterial medications are targeted at the specific subunits of the bacterial ribosome, because if you can shut down the bacterial ribosome, you can shut down the bacterial protein production. Next is the ER. The ER is shown in the image on the top right, which is an animal cell. The ER is in a blue color surrounding the nucleus. It is also shown in a larger format in the bottom right. There is another transmission EM showing the tubes of the ER as well as the beads, which represent the ribosomes of the rough ER. ER with ribosomes is known as rough ER, and without ribosomes is smooth ER. The ER is a network of membrane-bound tubes that is adjacent to the nucleus. Proteins are made in the ribosomes found on the rough ER, and within the ER, lipids are made. Additionally, within the ER, carbohydrates are metabolized and calcium is stored. The ER makes several necessary proteins, including trafficking proteins, secretory proteins, and membrane proteins. The ER can work through enzymatic reactions to detoxify the cell. Next, in the endomembrane system, we're going to cover the Golgi. In the image on the top right, there's an animal cell, and the Golgi is in a teal color. Looking at the image on the bottom right, the Golgi has two faces, the cis and the trans Golgi. The Golgi receives vesicles on its cis side. The cis side of the Golgi is closest to the ER, where it receives most of its vesicles. The trans face, or the trans Golgi network, or the TGN, is on the sending or the shipping side of the Golgi that sends vesicles out to the cell. While the ER is tubular, the Golgi is made up of flattened sacs known as cisterna. The cisterna closest to the ER is known as the cis-Golgi cisterna, and the cisterna known furthest from the ER is known as the TGN, or the trans-Golgi network. In the bottom right, there is a TEM image depicting the appearance of the Golgi cisterna. The Golgi is not only responsible for receiving and shipping of vesicles, it can also modify some of the content within those vesicles. As we move through the endomembrane system, we're going to look next at the lysosomes. In the figure on the top right, there's an animal cell with a lysosome depicted as a small purple ball. In the image on the lower right, the lysosome can be seen by EM as well. Lysosomes are membrane-bound sacs that are used to digest macromolecules. In the final image on the bottom, there is a diagram of lysosomes, which contain digestive enzymes that aid in breaking down macromolecules during digestion. Lysosomes can also receive vesicles that contain misfolded proteins or receptors, or have inactive proteins that need to be broken down. The final member of the endomembrane system are the vacuoles. These are usually found in plant cells, which is shown in the image on the top right. In the image on the bottom right, you can observe the central vacuole and the EM of the central vacuole. You can appreciate the total volume these vacuoles take up in a plant cell. They are large vesicles that originated either from the ER or the Golgi. They're necessary for food storage, water balance, ion balance, and this maintains all of these balances for the cell. The next internal set of structures that we're going to consider are metabolic compartments, the mitochondria, the chloroplasts, and the peroxisomes. In the image on the top right, there's an animal cell, and the cross section allows you to visualize the mitochondria in a yellow-orange color. In the lower right, there is a drawing of the mitochondria as well as the TEM of the mitochondria. We know that mitochondria have some genomic DNA, and they are the site for cellular respiration, which you will cover more in Bio 111. Because of cellular respiration reaction, which occurs in the mitochondria, mitochondria generate ATP, and because of this, they are known as the powerhouse of the cell. Again, in the top right image is an animal cell with the chloroplast shown in green. In the lower right, there is an image of the chloroplast as well as a TEM showing the structure of the chloroplast. Chloroplasts are usually found in plant cells, and they are the site of the photosynthesis reaction, again which you will cover in 111. Photosynthesis generates the power for the plant cells in conjunction with the mitochondria. Finally, the peroxisomes. These are shown in the animal cell on the top right as a blue ball and in the TEM image on the bottom right. Peroxisomes are a metabolic compartment with a single membrane that transfer hydrogen to an oxygen during certain enzymatic reactions, and this serves to detoxify the cells. Next, we're going to look at the bones of the cell, the cytoskeletal proteins. The cytoskeleton of the cell is a network of fibers that is found throughout the cell's cytoplasm and organizes the structures and activities of the cell. Some of these proteins are motor proteins, microtubules, microfilaments, and intermediate filaments. Let's look at the motor proteins. Cell motility and vesicle trafficking within the cell requires the interaction of the cytoskeleton with motor proteins. Motor proteins have feet that walk to their destination along the cytoskeletal track. A figure of a motor protein attached to a microtubule is shown on the right. The motor protein resembles an upside-down Y, with the feet attached to the microtubule and the top of the motor protein attached to the vesicle. This allows the vesicle to move throughout the cell to reach its final destination. Microtubules are another part of the cytoskeleton. Microtubules are structural hollow tubes that are made of tubulin dimers consisting of alpha and beta tubulin. These dimers form the tubes that shape and support the cell and serve as tracks for the motor proteins to move up and down. We'll also talk more about microtubules when we discuss cell division. In the figure on the bottom right, there is the microscopy image of an animal cell stained for tubulin. There's also a drawing of the hollow tube of tubulin, as well as the alpha and beta dimers that make up the hollow tube. Centrioles are found throughout the cell, again for shape and support. There is a pair of centrioles found in the centrosome. Centrioles are 9 sets of triplet microtubules in a ring formation. The centrioles are shown in the image on the bottom right. So as we've talked about microtubules, we've mentioned the centrioles. Now let's look at centrosomes. Centrosomes are a type of microtubules used by the cell. They grow near the nucleus and act as compression resistors that enable the cell to maintain its integrity and shape. The total of the centrosome is a pair of centrioles found in the centrosome, which is shown in the image on the bottom right. There are 2 sets of microtubules used for generating motion, cilia and flagella. These are extensions that project away from the cell wall. Cilia and flagella have very characteristic structures. The image on the bottom right shows a modal cilium, a cilia used for motor propulsion. The cross-section shows 9 pairs of outer microtubule doublets and a central microtubule pair in the center. These microtubules are held in place by other motor proteins called the dinins, as well as the radial spokes. Next, we're going to talk about a different type of structural protein, the microfilaments. These are different from microtubules because they are rods, not tubes. They are thin, solid rods that are twisted double chains of the protein actin. Microfilaments are necessary for supporting the shape of the cell by bearing tension. In the image on the bottom right, a cell is stained for actin, and there is a drawing of the twisted double chain of actin. Finally, we're going to talk about intermediate filaments. These are larger than microfilaments, but smaller than microtubules. They are not found in all cells, only a few animal cells. Intermediate filaments are specialized for bearing tension and aid in giving the cell shape. Finally, in this lecture, we are going to cover some of the external structures of a cell. Outside of the cell membrane, for plant cells, there is a plant cell wall made primarily of cellulose. The cell wall protects plants from damage, prevents major water intake or loss, and keeps the entire plant upright. There are some prokaryotes, fungus, and unicellular organisms that have cell walls as well as plants. Most cells have an extracellular matrix, or an ECM. These are made up of glycoproteins and carbohydrates that are created by the cell and used to attach to neighboring cells or the surface on which the cell is grown. An example of an ECM is collagen, which connects to the cell through fibronectins and integrins. These interactions are shown in the figure on the right. There are fibronectin molecules that are attached to the lipid bilayer of the cell, and these fibronectin molecules attach to the ECM molecule collagen. Finally, as we consider the external structures, we're going to talk about cell junctions. This is a physical contact between 2 cells that can be used for communication. Cell junctions are also known as tight junctions. These are both depicted in the image as well as in the drawing on the right. So let's use this video by way of reminder and review of what we've covered today on the cell.
Cells are the smallest living units of an organism. All cells have 3 things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
Today's lecture covered life's properties as a collaboration between the internal structures of a cell. We looked at prokaryotic and eukaryotic cells, as well as internal structures such as the nucleus, the endomembrane system, the mitochondria, the chloroplasts, the peroxisomes, the cytoskeleton, and external structures. If you have any additional questions, please feel free to email your professor or bring your questions to office hours. Additionally, you can bring your questions to class or reach out to your learning assistant who is assigned to this course. Now that the lecture is complete, combine your notes that you took during the lecture with notes that you took when you read the assigned chapter. As you've combined these notes and reviewed them, use the assisted reading questions as well as the questions at the end of each chapter and at the end of each chapter section to quiz yourself and prepare yourself for the quiz and the test on this material.